Welcome to Cell and Molecular Biology. Today, we are diving into one of the most fundamental and awe-inspiring processes in all of biology, translation. Imagine your cell as a bustling factory. The DNA is the master blueprint, locked in the manager's office. But the actual physical products, the proteins that do all the work, are built out on the factory floor. Translation is the incredible three-step assembly line process of initiation, elongation, and termination that reads those genetic instructions and physically constructs the proteins that keep you alive. By the end of this video, you will understand exactly how this molecular machine operates. To begin our journey, let us look at the very first step. Imagine you are building a massive, complex brick wall. Before you can start slapping on hundreds of bricks quickly, you must carefully lay the very first cornerstone. If this first brick is crooked, the entire wall will be unstable. In a living cell, translation initiation is exactly like laying that crucial cornerstone. It is the careful, highly regulated process of gathering the messenger RNA blueprint, the ribosome builder, and the very first amino acid building block. The small ribosomal subunit binds to the messenger RNA, guided by specific sequences. Then, a unique initiator transfer RNA, carrying formal methionine, binds directly to the start codon, which is almost always AUG. Finally, the large subunit locks everything into place. This step is absolutely critical because it establishes the correct reading frame, ensuring every subsequent three-letter word is read perfectly. Now that we understand what initiation is, let us look at the energetic cost of running this factory. Building proteins is one of the most biologically expensive processes in any living cell. The cell uses initiation as a major control checkpoint to ensure it only spends energy when absolutely necessary. The formula for this energy cost is E equals 4 times N. Conceptually, this comes from the fact that every single amino acid added requires the breaking of 4 high-energy phosphate bonds derived from ATP and GTP. In this equation, E represents the total number of high-energy phosphate bonds consumed. The variable N represents the total number of amino acids in that specific protein chain. The number 4 is the constant energy cost per amino acid. 2 bonds to attach the amino acid to its transfer RNA, 1 to bring it to the ribosome, and 1 to move the ribosome forward. As the protein length N increases, the total energy, E, increases linearly. Before we move on, let us see this with a concrete example. A bacterial cell needs to synthesize a molecule of green fluorescent protein to emit light. We are given that this protein consists of exactly 238 amino acids, and we are asked to find the total number of high-energy phosphate bonds required to build it. We chose our translation energetics formula because it directly relates protein length to energy cost. We start by writing E equals 4 times N. Next, we substitute our known variable N with 238. Multiplying this by the constant cost of 4 bonds per amino acid gives us our result. The final answer is 952 high-energy phosphate bonds. Physically, this means the cell must hydrolyze nearly 1,000 ATP and GTP molecules just to synthesize one single copy of this relatively small protein. This formula we just saw leads us to another important scenario. During a period of severe starvation, a cell has a depleted energy reserve containing only 1,200 available high-energy phosphate bonds dedicated to translation. We are asked to find the maximum length of a single protein chain the cell can synthesize. We use the same energetics formula, but this time we are solving backwards for the protein length, N. We substitute the known total energy, setting 1200 equal to 4 times n. To isolate n, we divide both sides of the equation by the constant value of 4. Dividing 1200 by 4 gives us our result. The final answer is 300 amino acids. This tells us that under these specific starvation conditions, the cell can only afford to fully translate a protein up to 300 amino acids long, forcing it to prioritize short survival proteins. Now that we have calculated the energy costs, let us visualize the physical assembly we just discussed. Here you are looking at the translation initiation complex. Notice the horizontal strand at the bottom. That is the messenger RNA blueprint, reading from the 5' end to the 3' end. You can see the small 30S ribosomal subunit already docked onto the messenger RNA, guided by the Shine-Dalgarno sequence. Right in the middle, the initiator transfer RNA is perfectly aligned with the AUG start codon, carrying the very first amino acid. Finally, look at the large 50S ribosomal subunit coming down from the top. The arrow indicates the final step of assembly, where this large subunit clamps down, creating a fully functional, intact ribosome. This diagram beautifully illustrates how the cell ensures the reading frame is perfectly set before spending any energy on elongation. With the initiation complex fully assembled, we move to the next major phase. Once the cornerstone is laid, the construction crew gets into a rhythmic, repetitive groove. Translation elongation is this exact repetitive cycle happening inside the cell. The ribosome acts as a molecular factory assembly line. It consists of 3 main steps that repeat over and over. First, a new transfer RNA carrying an amino acid binds to the empty A site of the ribosome. Second, the ribosome catalyzes the formation of a peptide bond, transferring the growing chain to the new amino acid. Third, a process called translocation pushes the ribosome exactly 1 codon forward along the messenger RNA, opening up the A site for the next incoming transfer RNA. Elongation is where the actual physical product is built. The speed of this phase is critical. If a bacteria encounters a toxin, it must rapidly elongate defensive proteins to survive. Understanding how this assembly line works naturally brings us to the question of speed. How fast does this factory operate? We can model this using a simple kinematic equation. T equals N divided by V. Conceptually, this is just like calculating the time it takes to drive a certain distance at a certain speed. Here, the variable T represents the total time in seconds required to completely translate the protein. The variable N is the total number of amino acids in the protein chain, which acts as our distance. The variable V represents the elongation velocity, measured in amino acids added per second. This equation tells us that the total time T increases if the protein length N is larger, but the time decreases if the ribosome works at a faster velocity, V. This helps biologists understand the physical time constraints a cell faces when expressing genes. Let us apply this kinetics formula to a real biological example. A human cell is actively translating a molecule of hemoglobin to help carry oxygen in the blood. We are given that the specific polypeptide chain is 146 amino acids long, and the eukaryotic ribosome operates at a steady velocity of 5 amino acids per second. We are asked to find how long it will take to finish this chain. We choose our translation time formula because it links length, speed, and time. We substitute the known variable N with 146 and the variable V with 5. We then perform the division to find the total number of seconds the ribosome spends moving down the messenger RNA. The final answer is 29.2 seconds. This physical interpretation shows that it takes roughly half a minute for a single ribosome to completely synthesize a hemoglobin subunit. Now that we have calculated time, let us look at a scenario where we need to find the speed. A fast-growing bacterial cell synthesizes a massive structural protein that is 3,000 amino acids long. The cell completes this entire process in exactly 150 seconds. We are asked to determine the elongation velocity of the bacterial ribosome. We start with our standard time equation, T equals N divided by V. We substitute our known time of 150 seconds and our length of 3,000 amino acids. To solve for the velocity, V, we multiply both sides by V to remove it from the denominator, and then divide both sides by 150. Dividing 3,000 by 150 gives us our rate. The final answer is 20 amino acids per second. This reveals that bacterial ribosomes operate significantly faster than eukaryotic ones, allowing rapid adaptation. Having calculated the speed of elongation, let us visualize the physical cycle driving it. Here, you see the translation elongation cycle broken into two main phases. In the top half, look at the ribosome's three chambers, the E, P, and A sites. A new transfer RNA carrying a single red amino acid enters the empty A site on the right. Notice the arrow pointing from the long polypeptide chain in the P site over to the A site. This represents the ribosome catalyzing the peptide bond, transferring the entire growing chain onto the new amino acid. Now look at the bottom half. The large arrow labeled translocation shows the entire ribosome ratcheting exactly one codon to the right along the messenger RNA. This movement shifts the empty transfer RNA into the E site, where it exits, and perfectly positions the growing chain back into the P site, leaving the A site empty and ready to repeat the cycle. We have seen how the protein is started and how it is elongated. But every good story must have a clear ending, and every assembly line must have a stop button. If a factory just kept adding parts endlessly, the product would be a useless, tangled mess. Translation termination is this final release process, ensuring the protein is cut free and the factory is dismantled. Termination occurs when the ribosome's A site encounters one of the three stop codons on the messenger RNA, UAA, UAG, or UGA. Unlike normal codons, there are absolutely no transfer RNAs that match these stop signals. Instead, special proteins called release factors enter the A site. These factors trick the ribosome into adding a water molecule to the end of the chain. This hydrolysis reaction severs the chemical bond, releasing the newly born protein into the cell. Now that the protein is released, we must ask a critical question. Is it built correctly? The accuracy of the entire translation process is known as fidelity, and it is ultimately judged by the final completed product. Without proper termination, ribosomes would cause massive traffic jams. But even with proper termination, if errors were made during the rapid elongation phase, the released protein might misfold. A misfolded protein is not just useless, it can be toxic and cause severe diseases. Therefore, the overall error rate is a critical factor for cellular health. The cell must balance the need for rapid elongation with the absolute necessity of high fidelity, ensuring that the final sequence of amino acids perfectly matches the original genetic blueprint. To truly understand the challenge of fidelity, we must look at the mathematics behind it. The formula for translation fidelity is P equals 1 minus epsilon, all raised to the power of n. Conceptually, this is a basic probability equation. 1 minus epsilon represents the chance of getting a single step right. Because every step must be correct to get a perfect protein, we multiply that single step probability by itself n times. In this formula, the variable p represents the overall chance, ranging from 0 to 1, that the completely terminated protein has absolutely no mistakes. The variable epsilon is the error rate at a single step. The variable n is the total number of amino acids. Because the term inside the parentheses is less than 1, raising it to the power of n means that as the protein gets longer, the probability of it being perfectly error-free drops exponentially. Let us see how this probability plays out for a typical protein. A ribosome is translating a moderately sized enzyme that is 400 amino acids in length. The ribosome has a standard error rate of inserting 1 incorrect amino acid per 10,000 steps, meaning epsilon is 0.0001. We are asked to calculate the probability that the completely terminated protein is absolutely perfect. We use our translation fidelity probability formula. We substitute the given error rate and the protein length of 400 into the equation. We first perform the subtraction inside the parentheses to find the probability of a single step being correct, which is 0.9999. We then raise this to the power of 400. The final answer is approximately 0.9608. Physically, this means about 4% of these synthesized enzymes will contain at least one mistake. Now that we have seen a standard enzyme, let us look at an extreme biological case. A muscle cell is translating titin, the largest known protein in the human body, with a staggering length of 30,000 amino acids. Assuming the same standard error rate of 0.0001 per step, what is the probability that a fully terminated titin molecule is completely error-free? We again use the translation fidelity formula to demonstrate the exponential decay of accuracy. We substitute the massive protein length and the error rate into our equation. We calculate the single correct step probability and raise it to the massive power of 30,000. The final answer is approximately 0.0498, or just under 5%. This is a profound physical result. It means over 95% of these giant proteins are born with at least one mutation, forcing the cell to rely on helper proteins to fix them. Having calculated the fidelity of the final product, let us visualize exactly how that product is released. Here, you see the mechanism of translation termination. On the left side, notice the red UAG stop codon positioned perfectly in the A site. Because no transfer RNA can read this, a specialized release factor, labeled RF, enters the A site instead. Look closely at the water molecule being introduced. This triggers the hydrolysis reaction. Now, look at the right side of the diagram to see the aftermath. The chemical bond has been severed, and the fully completed free polypeptide chain is floating away to fold into its functional shape. The empty transfer RNA is ejected, and the entire factory dismantles. The large 50S and small 30S ribosomal subunits separate from the messenger RNA, ready to be recycled for the next round of translation. We have covered the entire incredible journey of protein synthesis. Let us review the key takeaways. First, initiation is the crucial cornerstone. It sets the exact reading frame and acts as a strict energy checkpoint before the cell commits resources. Second, elongation is the rapid, repetitive assembly line that physically builds the polypeptide chain, dictating the speed of cellular response. Third, termination is the definitive stop signal, using release factors to free the completed protein. We also learned that this entire process is massively energy dependent, costing 4 high-energy phosphate bonds for every single amino acid added. Finally, we saw the mathematical reality of fidelity. As proteins get longer, the probability of a perfectly error-free product drops exponentially. You now understand the mechanics, kinetics, and energetics of how your cells build the molecules of life.